Here, I'm ready to go through Biology Paper 1, B3, Module Infection and Response. So I've looked at mark schemes. I'm just going to try and help you with the key content you need for this unit. Are we ready? So you'll need to know what a pathogen is, and that a pathogen is basically a microorganism that can cause an infectious disease. Infectious diseases, which the type of pathogens you need to know about are bacteria, viruses, protists, or fungi. Most of these groups of microorganisms are not harmful and not pathogens. Only a small percentage is a pathogen. Remember that the key important thing is that a pathogen are these microorganisms that can cause infectious disease. They can all be spread by water, contact, or air, and different types of microorganisms have different methods. Here you can see the basic structure of what a bacteria might look like. Remember, the DNA is free inside the cell, it has no nucleus. A virus doesn't even have a cell membrane or any cytoplasm. It is literally a protein coat surrounded by genetic material. What a protist look like, that looks like a more similar cell there. You can see it's got a nucleus and it's also got mitochondria in that cell given there. And a fungi again is eukaryotic because it has a nucleus. You can see it has a cell membrane and actually a cell wall as well, vacuole inside the center of it, and cytoplasm. So that basic structure from B1 is important in this unit. The types of bacterial diseases you need to know about. The first one is specifically Salmonella. In general, bacteria reproduce rapidly when they're in the body, and they may produce toxins. And these toxins damage the tissues and make us feel ill. Our immune system has to defend us against this, and we'll look at that in a bit. But Salmonella is basically what you get when you get food poisoning. There are other types of bacteria that can cause food poisoning, but Salmonella is one of the key types that you might catch. It's spread by bacteria in ingested food. If you prepare food in unhygienic conditions, for example, uncooked chicken. In the UK, to prevent this, the poultry, like chicken and turkey and things like that, excuse me, are vaccinated against Salmonella in order to control the spread. If you do get Salmonella, the symptoms there are shown in that diagram. You have fever, abdominal pains, vomiting and diarrhea, and that's all caused by the growing bacteria and the toxins that they're making. The other bacterial disease you need to know about is gonorrhea. Again, gonorrhea reproduces rapidly inside the body and produces toxins. It is actually a sexually transmitted disease with symptoms of thick yellow or green discharge from the vagina or penis and pain on urinating. It's caused by a bacterium which was easily treated with penicillin. However, gonorrhea then became resistant to penicillin, and now you can get gonorrhea and you need to use different antibiotics in order to treat it. Gonorrhea is spread by sexual contact, and the spread can be controlled by treatment with the right kind of antibiotic, and you can use barrier methods like condoms just to prevent the spread of the disease. In terms of viral diseases, this human viral disease of measles, you need to know what the symptoms are and how you can prevent that spread. So viruses reproduce differently to bacteria. When viruses reproduce, they have to be inside cells. So they get into the cells in the body, and this is what causes the cell damage. When they reproduce inside a cell, they actually damage the cell as they leave. Measles is a viral disease which affects humans, and you can get a fever and a red skin rash as a result. And it's a serious illness that can be fatal if complications arise. Lots of reasons, including that fatality reason, is a key reason why we get vaccines. And so you have vaccines against measles. The measles virus can be spread by inhalation of droplets from sneezes and coughs. So to prevent the spread of measles, you have to prevent the spread of sneezes and coughs. For example, using a tissue and disposing of the tissue in the bin, or covering your mouth, or making sure that you've washed hands thoroughly. If you've coughed onto your hands, can all be methods of avoiding spread, because you have to avoid it being inhaled. HIV is another human viral disease, and initially you will end up with flu-like illness, but then the complications will develop from that if it's not treated. It's recently been very successfully controlled with antiretroviral drugs, which can actually keep the HIV virus under control. The virus works in a very clever way by attacking the body's immune cells. So those cells that are there to prevent you getting ill are actually attacked by the HIV virus. With late stage infection, this is what caused most of the symptoms that you would see when you develop AIDS. And AIDS occurs because the immune system becomes so badly damaged it can't deal with other infections or cancers that may occur in the body. HIV can be spread by sexual contact or exchanging body fluids such as blood. So, for example, drug users sharing needles, that would be a way that you can spread HIV. So, avoiding the spread of it in that way, you can think of lots of strategies for that. You also need to know that plants can have viruses and pathogens too. And the viral disease you need to know about is TMV. So TMV stands for tobacco mosaic virus, and that's a plant pathogen that affects many types of plants, including tomatoes. It's called mosaic virus because it gives a distinctive mosaic pattern, so that sort of Roman mosaic pattern that you might see around and about. And that's discoloration of the leaves in that pattern. So you get green parts where there's chlorophyll and no green parts where there is less chlorophyll. This means less light can be absorbed in those cells that don't have chlorophyll in them, and you get reduced photosynthesis. So the plants make less glucose and there are fewer proteins for growth. So TMV, you'd have slow growth of a plant because there are less chlorophyll areas with the ability to photosynthesize. Another plant disease is a fungal disease called rose black spot. And fungi basically can grow anywhere in parts of the body or a living organism which are moist and warm and have enough nutrients. And removing one of these factors often helps to control the spread of fungal diseases. So rose black spot is a fungal disease which affects plants and causes purple or black spots to develop on the leaves. They turn yellow and drop early. Again, less chlorophyll is there, so less light can be absorbed for photosynthesis, and you make less glucose, and there are fewer proteins for growth. Because it is a fungus, it can be spread by water or wind. So rose black spot can really be treated by using fungicides, which kill fungi, or removing and destroying the affected leaves. And by destroying, it means properly killing them. So not just burying them in the ground, but actually burning them or something similar. The protist disease that you need to know about is malaria, and the pathogen that causes malaria are these small protists that actually live inside a mosquito. So the mosquito is a vector of this malaria protist, and because it is not harmed by the protist itself, it carries the pathogen, but it's not affected by it. So if an adult mosquito, which has the malaria protist inside it, bites a human, the protist enters the blood. The protist then reproduces in the liver, and that causes the liver to become damaged. Obviously, the protist is then in the blood. If you're bitten by another mosquito, that protist will go into that mosquito as well, and hence the mosquitoes are the main method of spread of the malarial protist. Malaria can cause recurrent episodes of fever and can be very fatal. In order to prevent malaria, you need to prevent the numbers of mosquitoes that there are, as well as preventing the mosquitoes biting you. So you can stop mosquitoes from breeding if you drain the swamps where they breed. You can kill the mosquitoes using insecticides. You can use mosquito nets and insect repellents to reduce chances of being bitten. You can actually vaccine, although it's not really massively effective as a vaccine. There are some vaccines for malaria, and you can take anti-malarial tablets, and they help to kill the pathogen should it become in your body. There are lots of ways that humans have to prevent all of these pathogens from infecting us, and they can be grouped into either specific defences or non-specific defences. So in terms of non-specific defences, it doesn't matter which type of pathogen is trying to infect your body. These are our preventative mechanisms for stopping them from getting in. So your skin is a physical barrier. If the skin is cut, a scab, which is basically a clot, is quickly formed to repair the barrier, and that uses the platelets in the blood. And the skin also produces antimicrobial secretions, which kill pathogens. So that's just really small amounts of kind of chemicals that we secrete onto our skin, which keeps it healthy and helps to kill pathogens. The nose with hairs and mucus trap dust and any microbes that you inhale through your nose. The trachea and bronchi also secrete mucus to trap pathogens and have ciliated cells. There you can see a diagram. Ciliated cells have these cilia, which are hair-like protrusions on them, which help to move the mucus out of the lungs. The stomach contains hydrochloric acid, which kills bacteria. So you can see there are a lot of links here to the B3 specific defense systems, non-specific defense systems rather, and B2. In terms of our specific defense system, we're really thinking about our immune system. So if our pathogen does manage to get through our skin or we inhale it, then it's entered the body, and this is where the immune system tries to destroy the pathogen. The key cell in the immune system are white blood cells, and they help us to defend in 3 ways. First way is phagocytosis. That term means the engulfing and digestion of pathogens. So that is a term you have to associate with white blood cells. And there you can see a diagram of phagocytosis. So the white blood cell actually is called a phagocyte in this sense. And it will engulf the bacterium and it will secrete enzymes into the bacteria and digest the bacteria and destroy it. So that's one way white blood cells help us to defend against pathogens. Another way is that they produce antibodies, and they are specific to the outside proteins on a pathogen called an antigen. And these antibodies will stick onto the antigen and cause the pathogens to clump together. This prevents their growth and also increases the rate of phagocytosis. I'll go into a bit more detail about this on the next slide. And the third way white blood cells help to defend pathogens is producing antitoxins. And these are basically chemicals that destroy the toxins that are released by pathogens. So remember, bacteria release toxins, and so white blood cells releasing antitoxins will stop those toxins from being so harmful. So really how this works in developing our immunity relies on those white blood cells with those antibodies. In this kind of cartoon of what happens if you get infected with a pathogen, you can see there are some really important keywords in here to be aware of and be able to use. So the first square says that all pathogens have specific antigens on their surface, and they're unique to that microorganism. So for example, if it's salmonella, it will have a salmonella antigen on its surface. All white blood cells also have a specific antibody this time on their surface that can attach to these antigens. And these antibodies are Y-shaped proteins that white blood cells produce that are specific to the antigen on any pathogen. If your body is infected with a pathogen, then one white blood cell with the right antibody, the one that is a match to the antigen, will be found. It might take a bit of time to find this white blood cell because you basically have to look through all your white blood cells to find the matching antibody. But once you do find it, you will start to get better from that disease. While you're looking for the white blood cell with the right antibody, it might be that you do get symptoms and feel ill. When you found the white blood cell with the right shaped antibody to that antigen, that white blood cell copies itself by mitosis to make many copies of the white blood cell and lots of copies of that antibody, which is specific to that antigen. And the antibodies then go attaching to any antigen that it comes across, and this causes the pathogens to be destroyed. One of the ways it destroys the pathogens is by causing them to clump together to make it easier for phagocytosis to happen. Now, the key thing and where this leads to immunity is that this white blood cell with the specific antibody to the antigen of the evading pathogen will be remembered. It will become what is called a memory cell. So if you're infected with that same pathogen again, then you can quickly find that white blood cell and release lots of antibodies straight away. And that's what we call immunity. This time round, if you get infected again with the same pathogen, you don't get symptoms, you are immune. And that's how vaccination works. It relies upon our immune system. So vaccination is basically preventing an illness by giving someone an antigen. And in order for vaccination to work, we need lots of people to take the vaccine, and that's called herd immunity. If there isn't above sort of 70, 80, 90% of the population immunised, then that disease can spread around the population. We like to have a large proportion vaccinated for this to work. Vaccination involves introducing small quantities of dead or inactive forms of pathogen into the body, and those pathogens will carry those antigens. This stimulates the white blood cells to produce antibodies to the antigens. This means when you've gone through your vaccine, you've had it, you're going to make memory cells. So if the same pathogen re-enters the body, your memory white blood cells can respond very quickly. They can make the correct antibodies, quickly killing the pathogen, so they don't produce a large enough, and not enough people in the population will have that pathogen and it can't spread. Infection is therefore prevented. And here you can see what it might look like after your first injection of a vaccine or an infection by a pathogen, and the second injection of your vaccine or in the actual infection itself. So you can see the concentration of antibodies in your first injection of a vaccine actually starts to rise and it goes up, and then it starts to drop off. If you're exposed to that pathogen or you get a second injection, a booster vaccine, then you can see that that rise of antibodies in your blood is much higher, it's much faster, and it will stay at a high level for longer. What that enables you to do is to be protected against the pathogen, which is obviously in the population and circulating around. So those three things about your antibody concentration after a second injection or an infection following a vaccine is that the antibody concentration is higher, it's faster increase, and it stays at a higher level for longer. Okay, if you are ill with a disease, you do need your immune system to destroy that pathogen in order to get better. However, there are some treatments that you can take which might help. Antibiotics are one of them. The antibiotics such as penicillin, they're medicines that can help to cure bacterial disease by killing bacteria in the body. And they only work on bacteria. They do not kill viruses. The use of antibiotics has greatly reduced deaths from infectious bacterial diseases, but there is nothing similar to kill viruses. It's very difficult, in fact, to develop drugs that kill viruses without also damaging the body's tissues because the viruses reproduce inside living body cells. So many drugs would kill the virus, but also kill those body cells. So you're not actually going to treat the virus very, very well, and you're going to have lots and lots of side effects. Specific bacteria, though, should be treated by specific antibiotics. With the emergence, though, of strains resistant to antibiotics, like we talked about gonorrhea earlier, we have to be very considerate about when we use antibiotics now, because bacteria have a habit of developing random mutations that makes them not immune, do not say immune, that makes them able to survive and reproduce. So bacteria cannot be killed by antibiotics. If you have a painkiller, they will only relieve the symptoms of the disease. They can't be used to kill the pathogens. They do not kill pathogens. They just help you to relieve the symptoms. So be careful. Painkillers relieve symptoms. Antibiotics will kill bacteria. They don't work on viruses. There are 3 drugs that you need to know the origins of and where they were developed. And the 3 drugs are the heart drug digitalis, which comes from foxgloves, and that's used to regulate heartbeat. The painkiller aspirin, which comes from willow tree bark. Penicillin, which was discovered quite by accident by Alexander Fleming from the penicillium mold. And those 3 drugs and their origins, you need to be clear about their uses and who discovered them and where they've come from. Most new drugs are synthesized, though, by chemists in the pharmaceutical industry, and they will synthesize drugs now. Some of the time, these are from plant extracts, so they still might look to the living world to look for drugs and treatments. However, sometimes they can be computer modeled and they can generate a completely new drug just using computer models. In a drug trial, there are some key phases. Drugs need to be really rigorously tested and trialed because we need to look for toxicity, that means how poisonous they are, their efficacy, that means how well they work, the dose, how much we need to give patients, and any side effects that may occur. I'd probably say all drugs have some side effects, and we need to know what those side effects are and how severe they are. So there are key stages in clinical trials, preclinical trials, which is basically the computer analysis of the drug and the discovery. And there'll also be some cells tested there. And then stage 2, which is animal testing, that is still a preclinical trial. Then we go on to clinical trials, and you can see here there are 3 phases shown. So you start with a small number of healthy volunteers, and then a small number of patients, so they would be people with the disease, and then a larger number of patients, so they're people with the disease who you're trying to treat, and then you get it approved and licensed for use. Let's just have a quick look at those stages in a bit more detail to see how they link to testing for their toxicity, efficacy, dose, and side effects. So preclinical testing is done in a laboratory using cells, tissues, and live animals. We would call all of those stages preclinical testing. And drugs are mainly tested at this stage for their toxicity. You're not going to be able to test on a living animal if a drug for a condition that humans have will work on that animal very well. It could be things like cancer, because it's a similar mechanism in animals, could be tested for at this stage. But really, you're looking for toxicity and maybe a little bit of some sort of efficacy if there is a disease that is similar across the animals you're testing. Clinical trials, though, is when you use healthy volunteers and patients, so they are people with the disease you're trying to make a drug for. And these stages, the clinical trials, are all used at very low doses of the drug, first of all, to give healthy volunteers. And that's the very start of a clinical trial. You're looking for toxicity and side effects at that stage. If the drug at that stage passes and is found to be safe, you will do further clinical trials to test the efficacy and find the optimum dose for the drug. And you would do those on the patients with the disease. So you would start to look at how well does that work in patients with the disease. You're still monitoring side effects at that stage as well. When you do clinical trials, whether it's on the healthy volunteers or patients, you would use double-blind trials. That is when the doctor or the patient don't know if they are getting the real drug or a placebo. And the placebo looks exactly the same as the real drug, but it just hasn't got that chemical ingredient, which is the drug itself, which will make a sort of the disease or give side effects or whatever. This helps to remove the bias. So the doctor, who might be invested because they're employed by that company to test the drugs, and they might be a bit biased if they knew they were giving patients the drug. So this removes any bias by using double-blind trials. And when a drug has passed all the stages of the trial, it is peer-reviewed. That means scientists look at how well the drug's trial has been carried out, make sure it's been done fairly and effectively, and looks at all of the methodology used and make sure that it's a robust trial. And then if everything looks fine, it's licensed and approved. Side effects, though, are still monitored. So if you get this drug from a doctor, for example, and you went and told him about side effects, they would still record that somewhere and share that data with the drug company. Okay, that's it really for B3. It's not a massive unit, but there's a lot of important recall in it. Recall of the types of different types of diseases and how they're spread, as well as recall of the different types of drugs and that drug's trial. So make sure if you need to go over it again, practice a few exam questions, and of course there's things like Quizlet you can do to help with the knowledge. Good luck.